In this lecture, we'll talk about epigenetics. The term epigenetics refers to heritable changes in the gene expression, active versus inactive genes, that does not involve changes to underlying DNA sequence, a change in phenotype without a change in genotype. So in this epigenetics, there is no involvement of change in DNA sequence. So DNA sequence will remain the same, but there will be change in phenotypic expression. So phenotype may change. So there is a change in the phenotype without a change in genotype. So DNA sequence will remain the same, but the phenotypic expression will change. So this is epigenetics. Epigenetic change is a regular and natural occurrence. So this is a regular process and also a natural process, but can also be influenced by several factors including age, the environment, lifestyle, and disease state. Even nutrition also, it can affect the epigenetics. So we'll learn some of these examples. So what is epigenetics and what role does it play in modulating the gene expression or changes in the our body? C.H. Weddington coined the term epigenetics to mean above or in addition to genetics to explain differentiation. So there were several questions how which can be explained only by epigenetics, like how do different adult stem cells know their fate? Myoblasts can only form muscle cells, keratinocytes only form skin cells, hematopoietic cells only become blood cells, but all have identical DNA sequences. Though they have the identical DNA sequences, but these cells differentiate into different types of cells. And how a stem cells has capability to differentiate different types of cells also. So these are epigenetics can explain all these phenomena. Modern definition is non-sequence dependent inheritance. Basically, it's not related to DNA sequence. How can identical twins have different natural hair colors? Identical twins should have everything same, but sometimes when they grow up, they may have different features like different hair colors. How can a single individual have two different eye colors? How can identical twin litter mates show different coat colors also, like in mice? Identical twins can have different coat colors. How can just paternal or maternal traits be expressed in offspring? This is called genetic imprinting. How can female express only one X chromosome per cell? So these are some of the questions which are now answered by epigenetics. How can acquired traits be passed on to offsprings? If some traits are acquired by parents, they can be passed on to offsprings. Some changes in gene expression that are in fact heritable. So this is just a picture showing identical twins. They are the identical twins, but they have different hair colors. So this coat color genetically identical agouti mouse litter mates. So they have different. Some are yellow and some are dark brown. So have different coat color, but they are genetically identical. They should ideally should have the same coat color, but they have different coat colors. So this can be explained by epigenetics. So epigenetic mechanism involves three things. Important one is DNA methylation, another one is histone modification, and the third one is RNA interference. So all these three leads to change in gene expression levels, and by modulating the gene expression levels, they carry out the epigenetics changes in the body. DNA methylation. Here the cytosine DNA methylation. In this, the cytosine residue undergo methylation. Here, this is cytosine by with help of DNA methyl transferases. Methyl group is transferred here, this particular carbon, and the DNA gets methylated. So the brown one, as you can see, methylated DNA, it gets, once DNA gets methylated, it get compressed. And when DNA gets unmethylated, it becomes more relaxed now. And DNA unwinds, it become more relaxed. And then once it get relaxed, then the gene expression or transcription will occur and the protein will get expressed. So this, once the DNA means unmethylated DNA, it undergoes it can transcription occurs when the DNA is unmethylated. Here we can see in this example, the brown one is methylation. Similarly, just like DNA methylation, acetylation of the histone protein, these protein which are again, so this DNA is wrapped around. So these histone protein undergo acetylation. So DNA gets methylated and the histone protein get acetylated. So in case, so here is the example here. The yellow one is acetylation, this histone protein is getting acetylated, and the brown one is DNA methylation, shows methylation pattern in the genome. Suppose here is the exon A, here is the exon B here now. So once, as you see here, there is a more of acetylation and less of DNA methylation. So you can say this is hypomethylated, less methylation, so this hypomethylated. In this, then DNA is more relaxed and the transcription takes place. Here you can see the transcription takes place. But when the same DNA sequence gets hypermethylated, you can see the brown one. So this particular exon 1 portion, in this one, hypermethylation occurs, DNA gets methylated and the histone protein gets deacetylated. So acetyl groups from the histone is removed. In this particular situation, now the transcription will not occur, it will be inhibited. So this is hypermethylation. So it means hypomethylation, DNA transcription occur. When it gets hypermethylated, DNA transcription doesn't take place. So protein gene expression will be repressed. So this is the structure shows. Here is this blue, light blue is the DNA double helix, and these are the histone protein around which the DNA is double helix DNA is wrapped. So these tails, these are histone tails. So these histone tails get acetylated. Amino acid, so lysine, particularly the lysine residue in the histone protein gets acetylated. So here's the example when the histone are unacetylated. When the histone are unacetylated and DNA is hypermethylated, chromosome is in compressed state. It's more compact. But when histone protein get acetylated and DNA get unmethylated, you see a more relaxed structure, and then transcription will take place. The acetylation of histone tails promote loose chromatin structure that permit transcription now. So acetylation of histone protein promotes the loose chromatin structure that permits transcription. So these are the different histone protein S2A, S2B, S3, H4. So all of these can get acetylated, acetylation group, acetylation group. S3 also get methylated also. So this slide just shows the histone modification of histone protein, how it can influence the chromatin structure. So DNA methylation and protein and histone acetylation is also known as the epigenetic code. The DNA methylation, methyl marks added to certain DNA bases repress gene activity. So DNA methylation or the hypermethylation leads to gene repression or inhibition of the gene activity, or the histone acetylation will also. Histone acetylation and the DNA hypomethylation will lead to transcription. So in case of DNA methylation, only the cytosine residue in the DNA undergo methylation with the help of DNA methyl transferase enzymes and presence of SAM, adenosyl methionine. So there is a this is cytosine residue. This is cytosine with the help of DNA methyl transferase. Methyl group is transferred in the this 5 carbon. So this is cytosine methylation. So this is the same thing here. This 5-methyl cytosine residue in the DNA, in the structure you can see here. So these are the methylated cytosine residue in the DNA. So the cytosine methylation maintains inactive condensed chromatin state. It maintains the inactive condensed chromatin state. Like here, when the DNA is hypomethylated, methylation is less, the DNA is more relaxed. So now the various transcription factors required, like RNA polymerase, they will be able to access the promoter region and the transcription will start. So in this situation, DNA is unmethylated. DNA is unmethylated and histones are acetylated. Histone acetylation and DNA demethylation or unmethylated DNA leads to transcription. Now in the next step, DNA methyl transferases come in presence of SAM. DNA cytosine residue in the DNA gets methylated. Okay. At the same time, when DNA is getting methylated, histone deacetylase will also come in picture and it will start deacetylating the histone protein. So now once the DNA methylation occurs and deacetylation of the histone protein occurs, DNA will start getting more compressed. Okay. So here now in this particular situation, DNA is hypermethylated and histone protein are deacetylated. So acetyl group from the histone protein is removed. So this is deacetylation, hypermethylation and deacetylation process. So what will happen now? Chromatin will become more compact now, and RNA polymerase and other transcription factor won't have access to the promoter regions, and the what will happen now, transcription will stop now, it will be inhibited. So this is how the DNA methylation and histone acetylation plays a role in regulation of the gene expression. So 5-methyl cytosine is found in heterochromatin region. So whatever the heterochromatin region in the genome, they are methylated. So DNA methylation and histone modification help to compartmentalize the genome into domains of different transcription potential. Euchromatin, in, euchromatin and heterochromatin. So euchromatin, high DNA, high histone acetylation, histone protein is acetylated here, and low DNA methylation, DNA is hypomethylated, and S3K4 is methylated. So this is now euchromatin here. So in case of euchromatin, expressions takes place. Expression of the protein will occur in this region. In case of heterochromatin, low histone acetylation, and DNA is hypermethylated. DNA is hypermethylated, is more compact also in structure. So chromatin is more compact, and S3K9 is methylated here in this case. S3K9. So in this situation, heterochromatin transcription will be inhibited. So how do this cytosine methylation is maintained during replication? Like suppose this is double-stranded DNA here now, both strands are methylated. It will undergo methylation. It will undergo replication. Then one strand will be methylated and the new strand will be unmethylated. So what will happen now? Then DNA methyl transferase come into picture. It will methylate the second strand, new strand also. Okay, so this can occur, or the second round of replication, passive demethylation can occur. In the other situation, in the second situation, the old strand also get unmethylated. So now both strands are unmethylated, and then in the next step, DNA methylation can occur in both the strands, and the methylation then will be cytosine methylation is then hence maintained during the replication process. This picture just to show that DNA methylation is a very important process in our body, and the enzyme which carried out this process, DNA methyl transferases, are very essential. So there are different types, different isotypes of DNA methyl transferase, like DNA methyl transferase 1, DNA methyl transferase 2, 3A, 3B, and 3,1. So it has been observed that DNA methyl transferase 1, when this particular gene is knocked out in mice, it has been knocked out in mice. When we remove this DNA methyl transferase 1, it is embryonic lethal. Means newborns are not born here. During embryonic stage, the fetus dies here in this situation. So it means these enzymes are very important. So like similarly, DNA 3A, 3B, they are all embryonic lethal. In DNA methyl transferase, no obvious effect was seen, but other DNA methyl transferases, they are so important that once we knock out these genes, these are removed from the body, then they become they're highly embryonic lethal now. So it means these genes are so important, and also suggesting that DNA methylation is a very important process in our body. The cytosine methylation in mammals plays very important role, particularly in regulating gene expression. It maintains chromosomal stability. It helps a lot in cell differentiation. We'll see that in the next slide. DNA imprinting, X inactivation. One of the X is inactivated. It can play very important in carcinogenesis also and aging process also. So this so DNA methylation plays very important role in our body. So the epigenetics can explain a very important question: the how is the diversity of cell types created and maintained in multicellular organisms. So this is very important question: how is the genome DNA sequence is same in all the cell types, but they perform different function and there is a lot of diversity. So how is the diversity of cell types created and maintained in multicellular organism? How it is created and then how it is maintained. So epigenetics can explain this phenomena. So when once the zygote undergoes cell division and gets differentiated and develop into adult, these differentiated cells, they become more restricted in their potential. Like in the during the zygote early development in the zygote, their cells in this zygote are highly totipotent. They have potential to get differentiated any type of cells. But as during the development, further stages of the development, these totipotent cells become pluripotent. So they have limited potential, but still they can differentiate many cell types. But still they are less ability as compared to totipotent to differentiate. And pluripotent, when again during further development, these pluripotent cells become multipotent. They have limited ability. They can differentiate into very few types of cells or tissues. But when during the adult stage, these cells then become unipotent, so they can differentiate only one type of cells. So this is how the cells become more restricted in potential to differentiate. So in this situation, DNA methylation plays a very important role. So DNA methylation differentiates totipotent embryonic stem cells from the unipotent adult cells. So this picture can explain actually how the DNA methylation can play important role in cell differentiation. So here is the pluripotent cells and unipotent cells. So both of these pluripotent cells and unipotent cells have a identical DNA sequence. So they're genetically they are identical, no difference in DNA sequence. But the how a pluripotent cell has more potential to differentiate, but the unipotent cell doesn't have much potential to differentiate into other cell types. So DNA methylation takes DNA methylation plays a very crucial role here. So this is again the DNA sequence of pluripotent cells, and this is DNA sequence of unipotent cells, and this shows the cytosine CG island. So you can say the cytosine residues present in pluripotent cell, and similarly these are the cytosine residue in the unipotent cell. It's identical sequence. This just to highlight the cytosine residues here. Now the difference occurs in, so here both the cells are same identical DNA sequence, but the pattern in DNA methylation is now different. So in case of pluripotent cells, there is no DNA methylation. So cytosine residues are not methylated. Okay, cytosine residues are not methylated. But in case of unipotent cell, cytosine residues are methylated now. So this methyl cytosine. So this is red color shows that DNA is now methylated. So in case of unipotent cell, DNA is hypermethylated. So it means some of the genes are not expressed in the unipotent cells, which are required in the pluripotent cells. So pluripotent cells, most of the genes required in development or differentiation are expressed continuously. But in case of unipotent cells, many genes which are required in development or differentiation, they are now silent now because of DNA methylation. So this is how the DNA methylation maintains the pluripotent cells and the unipotent cells. So now the reverse can also be true. Differentiated cells can become now totipotent. So nuclear transplantation demonstrates nuclear equivalence, like in the case of animal Dolly. In case of Dolly, differentiated memory gland cells were taken, and then they were used to create a exact copy of the sheep Dolly. So this is how Dolly was created, using the differentiated memory gland cells. So in that situation, differentiated cells became more totipotent later during the growth. So here generally the process is the natural process is totipotent. Cells become pluripotent, pluripotent become multipotent, and multipotent becomes unipotent. But in case of Dolly, in case of animal, this is animal cloning. Now these unipotent, which are differentiated cells can now become totipotent also. So this process also can be created now. So this process can occur via only when this unipotent cell become unmethylated. So once these become unmethylated, they retain the they they again retain the totipotency. So critical CPG sequence in the CPG islands near promoter region. So the regions, the CPG cytosine residue near promoter, particularly they are uh, highly prone to methylation in the CPG island. So this slide shows the genomic distribution of DNA methylation. How much genomic DNA is DNA methylated. So cytosine residue is the one which get methylated. So 4% of all cytosine are methylated. So 4% of all cytosine residue in the genome are methylated, and 70 to 80% of all CPG are methylated. So basically this particular region, CG region, you can say CG region. 70 to 80% of all CG regions are methylated. That is these are CPG island. Here you can see. So this slide shows actually methylation changes during development. So methylation plays very important role during development. So what happened actually here? Now this is the this is when the fertilization is occurring here. So this is the oocyte and the male sperm. The fertilization occurs. So this is a fertilized egg. Here this has now both genome from paternal genome also and maternal genome also. And this is one cell stage, two cell become four cell to eight cell, morula stage and blastocyst stage. So now this graph shows the methylation pattern now. So during oocyte and sperm before the fertilization, you can see DNA is more methylated here. DNA is methylated, but when the it become fertilization occurs and it undergoes cell division, one cell become two cell to four cells. Now you can see first the paternal genome. The paternal genomes undergo is become more unmethylated. So here you can see paternal genome is the first one which gets more unmethylated. You can see this region is the unmethylated now. And slowly in the one cell to two cell stage, maternal genome also start getting unmethylated. So you can see when the blastocyst at the blastocyst stage, both paternal genome and maternal genome are unmethylated. It means once this genome get unmethylated, the genes which are present in paternal genome and the maternal genome, they are unmethylated, and their thus expression starts then. Genes which are very important in the development, during development stage, they start getting expressed and play very important role in the development of the fetus. So this slide shows the demethylation of the paternal pronucleus in one cell embryo of the mouse, one cell embryo. So this is the ones after the fertilization, or the in the mouse after the fertilization, there is a this is paternal genome, this is maternal genome, and you can see the paternal genome is getting now demethylated. So demethylation is occurring in the paternal, and slowly this genome also will become unmethylated. So epigenetics also explain the phenomena of X chromosome inactivation on the presence of Barr bodies. X chromosome inactivation basically carried out hypermethylation of CG island. So this is two copies of X chromosome, one copy here, second copy here. Now the inactivation of one X, in female there are two X chromosome, but the one X chromosome is inactivated. So here X, this one is the upper one, which shows the red color. It is methylated now. So this become now inactivated X1, and the lower one, which is now unmethylated, hypomethylation, it is now it remains active. So the once after the X chromosome get methylated, hypermethylated, it becomes inactive. XIC region is the region, this particular region on the human X chromosome plays very important role in the inactivation of X chromosome. So characteristics of XIST region actually located in the XIC region of the X chromosome, transcribed only from the inactive X. So this expression of this particular region, particular gene, XIST gene, occurs only in the inactive X, not in the active X. So this inactive X, this particular gene gets expressed. This 20 kb cDNA with no ORF remain intranuclear surrounding the Barr bodies. The XI region responsible for 6 inactivation of the X chromosome. So how XIST gene silence the future inactive X? So this is expressed only from the inactive X chromosome. Here, this shows the expression of XIST. This is the XIST gene in the X chromosome. So this is expressed only, expression occurs only in the inactive X chromosome. So once this protein this gene get expressed, this protein actually coats the chromosome now. Once it coats the chromosome, it maintains the, it establishes the inactive state. Like in some case of genetic disorders, when there are extra copies of X chromosome, here in this case X chromosome, only the one X will remain active and other will become inactive. So this is now in case of female which has two X chromosome, one X chromosome is inactivated and one X chromosome will remain active. So this is in case of XX female. In some case of genetic disorder or syndromes, there are extra copies of X chromosome. Like in this X, there are 4X, 1, 2, 3, 4X and 1 Y chromosome. So in this 4X chromosome, out of 4X chromosome, 3 will become inactive here. So this is how the X chromosome is inactivated, and it leads to formation of Barr bodies. So these are the Barr bodies visualized by XIST RNA fish. XIST RNA phase was used as a probe, and it detected the 1 bar body in XX female and 3 bar bodies in XXXXY male. So inactive has X has unacylated histone 4 also. So the inactive X means it is hypermethylated, and thus at the same time histone protein H4 is unacetylated. So these are the different distinguished feature of inactive X chromosome and active X chromosome. Bar body formation occurs in inactive X chromosome. XIST expression occurs in inactive X chromosome. CpG islands are methylated. S3 is methylated histone protein. Histone tail acetylation is not present here. So these are some of the difference. In case of active X chromosome, bar body formation does not occur. XX expression does not occur here now. CpG islands are not methylated. S3 is not methylated, and histone tail acetylation is also takes place in case of active X chromosome. So histone acetylation occurs in active X chromosome only. So these are the differences between. Now it has been shown that epigenetics play a very important role in cancer also. The human body is prone to develop cancer cancer from a very early stage of life until the end of life. The human genome has several built-in tumor suppressor genes. Tumor suppressor genes are the genes actually which are involved in suppression of the tumor formation in our body. So whose protein products suppress the formation of tumor? It is important for these genes to continue expressing their tumor suppressor protein as long as the person lives. So as long as the tumor suppressor genes are expressed, it prevents the tumor formation in the body. One way these genes can lose their ability to make protective protein is through methylation. So, but once these DNA tumor suppression genes are methylated, they lose their ability to prevent cancer formation. So this is how the epigenetics can play a role in cancer progression in human body. So cancer was the first human disease to be linked to epigenetic. Studies performed by Feinberg and Vogelstein in 1983 using primary human tumor tissues found the genes of colorectal cancer were substantially hypermethylated compared with the normal tissues. DNA hypomethylation can activate. So DNA, what happens actually here? DNA hypomethylation can activate oncogenes. When the oncogene, these are the genes which are present in our body, but they are inactive. But once these oncogenes get activated because of DNA hypomethylation, they can cause cancer formation. It causes initiate chromosome instability, and at the same time, DNA hypermethylation initiate silencing of tumor suppressor gene. An accumulation of genetic and epigenetic factor can transform a normal cell into an invasive or metastatic tumor cell. So this is how DNA hypomethylation of oncogene and the hypermethylation of tumor suppressor genes can lead to cancer formation and cancer progression in the human body. So global histological pattern also found to correlate with cancers such as prostate, breast and pancreatic cancer. Epigenetic change can be used as biomarkers for the molecular diagnostic for early cancer also. So this is just to show that epigenetics can play a very important role in cancer progression in human body. So now this slide shows how the epigenetics changes can lead to differences in identical twin, monozygotic twins. Jabki monozygotic twins should be identical because they have identical DNA sequence. So they phenotypically also they should be identical. But during the development, epigenetics changes leads to difference in the monozygotic twins also. So like this particular graph shows that these are 3-year-old twins, and in 3-year-old twins you see the methylation pattern is almost same. This is DNA methylation is almost identical, but as they grow old, like this is 50-year-old same twins, you can see there is change in DNA methylation pattern. Methylation pattern is quite different between the twins, and this difference can leads to differences in the phenotype of the monozygotic twins. Similarly, this shows the acetylation now. This is 3-year-old twins. Acetylation of histone protein is identical in both the twins, but as they grow old, this is 50-year-old twin, the histone acetylation pattern also changes. So this shows that epigenetics also play a important role in the phenotypic expression of the uh, identical twins also. So monozygotic twins also can show differences in their phenotypic, mainly due to the epigenetics. So nutrition also play a very important role in the epigenome. So we'll talk about a case about the agouti genes in the mice, which are responsible for the coat color. So methylation of agouti genes in mice, methylation at the AY allele in sperm bisexual sequencing. If it is less methylated, 27% is methylated, its coat color is yellow. And if it is hypermethylated, it's a brown color. So what happened actually in this case of mouse? Experiment in mouse shows just how important a mother's diet is in shaping the epigenome of her offspring. All mammals have a gene called agouti gene. When a mouse agouti gene is completely unmethylated, it is unmethylated, its coat color is yellow here. This is unmethylated, coat color is yellow. And it is obese and prone to diabetes and cancer. So this is obese mouse and prone to various diseases like cancer and diabetes. When the agouti gene is methylated, as it is in the normal mouse, okay, the coat color is brown in color. So when it get methylated, the coat color is brown and the mouse has a low disease risk. Fat yellow mouse and skinny brown mice are genetically identical. So they are genetically identical, but because of the methylation, different in methylation pattern, one is prone to disease, one has yellow color and prone to disease, but here this is thin and healthy mouse, and it is a hypermethylated agouti genes. So when the scientists fed pregnant yellow mice, this particular mice, a methyl-rich diet, most of her pups were brown and stayed healthy for the life. So this is just to show that nutrition of the mother can change the epigenome of the offsprings. So when these mothers with the yellow coat were fed with high methyl-rich diet, their offsprings were in brown in color. So this shows that their DNA of the offsprings were methylated. These results shows that the environment in the womb influence adult health. In other words, our health is not only determined by what we eat, but also what our parents eat. So these were fed with normal diet. So more of the mice were actually having yellow coat color and prone to diseases. Here, when the mother was fed with modified diet with adding vitamin B12, folic acid, which are highly rich in methyl group, so their offsprings were healthy, brown coat color and very healthy. So this just to shows that epigenetics, nutrition of the mother can play a very important role in. So this is when the more methyl rich food is given to mother, their offsprings are healthy now. So just to summarize, so these epigenomes can play important role in inactivation, in DNA imprinting, heterochromatin maintenance, development control. It plays very important during development. Tissue specific expression can play a role in cancer and other diseases. So thanks for your attention.